following video will cover subtopic 1.3, membrane structure. The following is a list of understandings, applications, and skills that will be covered in this video. Before we go through membrane structure, we'll cover a little bit of vocabulary, as this vocabulary will become important in describing the components of the membrane. So molecules can be categorized according to how they interact with water. Molecules that are hydrophilic are substances that are attracted to water or will interact with water. Molecules that are described as being hydrophobic are substances that are not attracted to water, and they will look to stay away from water or will be repelled by water. And then you can have molecules that have both hydrophilic and hydrophobic properties, and these substances are described as being amphipathic. So they will, there are parts that will interact with water and parts that will not be attracted to the water. So when looking at membrane structure, we'll look at the individual components that go up into making that overall membrane structure, and we'll start with kind of the basic component, which is the phospholipid. So the phospholipid is what will ultimately be used to make up that cell membrane. The phospholipid has two parts to the molecule, which is often described as being the phosphate head. So it has a phosphate group at one end of the molecule, and this will represent, the phospho part of the name phospholipid. And then it will have a hydrocarbon tail or hydrocarbon tails. These will be the lipid part of the name. So when looking at these two components, the phosphate head, which is represented in this molecular diagram on the left. The phosphate head is found in this area that I've just circled, and will be hydrophilic in nature, so it will be attracted to the water. The hydrophobic tails are these long chains of carbon that will be repelled by the water or will not want to interact with the water. So these two components together make up that phospholipid. Diagrammatically, we can represent a phospholipid using the following. So we can have a circle that represents the phosphate head, and then we can represent those hydrocarbon tails with just some lines. And we can even simplify our drawings even further, just to represent a phospholipid as having that phosphate head, and then some squiggly lines to represent those hydrophobic tails or those hydrocarbon tails. So phospholipids, when you have a whole bunch of them together, will come together to form what is called a bilayer in water. And this is really due to the amphipathic properties of the phospholipid molecule. So if we have a look at the diagram, outlined in blue or black, is a phospholipid molecule. And the way the molecule will orient itself is that the positively charged phosphate head or phosphate heads will interact with the water found in that extracellular space. Because the hydrocarbon tails are hydrophobic, they will want to point away from the water or isolate themselves to the water. So the phosphate heads will arrange themselves so that they are all interacting with the water, and then the hydrocarbon tails will point away from the water. Where the bilayer gets formed is that in the lumen of the cell, where you will also have water molecules, those phosphate heads will arrange themselves so that they are pointing towards the water or interacting with the water. And then the hydrocarbon tails will now orient themselves so that they are interacting with other hydrocarbon tails that are adjacent to them, as well as the hydrocarbon tails from those molecules just above them. So in this way you form a bilayer, or two layers of phospholipids. So when mixed with water, those phosphate heads are going to be attracted to the water, and those hydrophobic tails are attracted to each other. And so as a result, they create that phospholipid bilayer, and the primary function of that bilayer is really to form a barrier through which ions and hydrophilic molecules cannot easily pass. Because of that hydrophobic core in the middle, something that is charged, we'll say such as sodium, or even water itself, has a hard time passing through because the water is not going to want to interact with that hydrophobic core and will be repelled by it. So it forms an effective barrier at keeping molecules on one side of the barrier, so we could say keeping things inside the cell, and then keeping things outside of the cell. There are alternative models for membrane structure. So when put in the water, an emergent property of phospholipids is that they will actually self-organize to keep their phosphate heads, we say, wet, and their hydrocarbon tails dry, meaning that the molecules will now orient themselves in these favorable positions where the phosphate head is going to interact with water and the hydrocarbon tails will try to hide from the water, essentially. So they will. So there's two types of structures that can be formed. There is the first arrangement, which is referred to as a micelle. This is where you have just a single layer of phospholipids, where it's a fairly small structure, and then you get all their tails kind of pointing towards the middle of this structure, excluding water from the center of it. You can also get a bilayer that is formed in what is referred to as a liposome. A liposome is going to have that bilayer, so you're going to have two layers of phospholipids, and in the center you will have an aqueous core that will contain water, as well as water on the outside, whereas in the micelle, it is just water on the outside, but no water on the inside. Also found in the structure of membranes, of cell membranes, are proteins, or what we'll refer to as membrane proteins. Membrane proteins carry out a number of different functions for the cell, which can include things like transport. So you can have protein channels, which carry out this process of facilitated diffusion, meaning that you'll have molecules that will move from a high concentration to a low concentration gradient naturally. And then you can have protein pumps, which will actively move substances from a low concentration to a high concentration through a process of active transport, which requires the use of energy in the form of ATP, whereas facilitated diffusion does not require ATP. And this will be covered in greater detail in the video for 1.4. Membrane proteins can also act or function as an anchorage point. So you can have those cytoskeleton filaments attach to the cell membrane, as well as the extracellular matrix. Proteins, membrane proteins can also act as receptors, where they can detect certain proteins binding to them, which would pass on signals into the cell. They can also act in cell recognition roles. For example, you can have receptors that will recognize certain molecules from foreign substances and then trigger an immune response. They can also have enzymatic activity, where they will initiate metabolic pathways, and then they can also join cells together. So if you have to have cells that are tightly joined, you can have what are called tight junction proteins, which will adhere these cells together. So membrane proteins can be largely classified into two types. There are integral proteins and peripheral proteins. So integral proteins are considered transmembrane, meaning that this protein will span the length of the membrane, of the cell membrane. So within an integral protein, there is going to be a hydrophobic portion, and that hydrophobic portion is going to be embedded in that hydrocarbon interior, or that hydrophobic interior. There is also going to be a hydrophilic portion to an integral protein, and this is going to project through the membrane and through the phosphate heads, and will interact with the aqueous solutions in the extracellular matrix or in the cytoplasmic region of the cell. So in the following diagram, we have an example of an integral protein. There is a portion of that protein that is embedded in that hydrophobic core, and then there is a portion that extends outside or beyond the cell membrane, which can interact with the aqueous environment. And then there is going to be a portion that extends to the inside of the cell, which can also interact with the aqueous environment within that cytoplasm. So one of the roles of integral proteins is that they can help transport substances across the membrane. So there can be an opening through the middle of the protein that can allow substances to move from the outside of the cell to the inside of the cell, or can even allow substances to move from the inside of the cell to the outside of the cell. The other type of category of membrane protein is the peripheral protein. So peripheral proteins are going to be hydrophilic on their surface, so the outer part of the protein is going to be hydrophilic and does not embed itself within the cell membrane. What they will do is sit on the membrane surface. And on the membrane surface, they can slide around easily and collide with each other, but basically they stay on the side that they are located on. So they cannot flip from one side to another. Most peripheral proteins you find associated or attached to integral proteins. And usually they can be involved in maintaining the cell shape or aiding with cell motility. But they also may be enzymes that catalyze reactions in the cytoplasm as well. So many different roles for these peripheral proteins. Another component of in membrane structure is cholesterol. So cholesterol is a common component of cell membranes. Cholesterol itself is categorized as a steroid, which makes it a type of lipid, but it's not considered a fat or an oil. It's kind of its own thing. The structure off to the right shows the molecular structure of cholesterol. So cholesterol is mostly hydrophobic, and when we say mostly, like the vast majority is hydrophobic. So pretty much the whole molecule is going to be hydrophobic. So there's a lot of hydrocarbon groups in there, a lot of carbon-carbon bonds, and so these will not interact favorably with water, making them hydrophobic. The only portion that is hydrophilic is the little hydroxyl group, or that OH group, at the end of the molecule. So this portion will be hydrophilic. So within the cell membrane, that hydroxyl group is going to be attached or interact with the phosphate head on the phospholipid. And then the rest of the molecule, the hydrophobic portion, is going to be embedded in that hydrocarbon, interacting with those hydrocarbon tails, or that hydrophobic core. So it is going to be positioned, where you'll find cholesterol is that it'll be positioned between the phospholipids in the membrane, and the amount of cholesterol that you find in a cell membrane will vary depending on the cell type. So some cell types will have more cholesterol in their cell membranes than others. It really depends on what the cell is, what the function of the cell is. So what does cholesterol do in the cell membrane? So what it'll do is that it'll make the phospholipids more tightly packed and can regulate the fluidity and flexibility of the membrane. So in the following diagram, we can find cholesterol here embedded in the membrane. So outlined is the hydrophobic portion, and then the little circular part is the hydrophilic portion of the molecule. So it will embed itself between those phospholipids, and it will help tightly pack those phospholipids together. It really kind of prevents them from moving around too much. So the analogy that goes with this in thinking about the role of cholesterol is that, you imagine a room full of people wearing fluffy sweaters. It's crowded, but everybody can still move around pretty easily now and can get to where they need to and is free to move about. But now think about what would happen if you sprinkle the crowd with people wearing Velcro suits. The people wearing the Velcro suits are going to keep those phospholipids from not moving around too easily. Like, some of them are not going to be able to move around as much. You're going to be tightly packed in a bit more and not free to move as you like. So those Velcro suits, you can think of as cholesterol in the cell membrane. So all these components together make up that membrane structure. And the model that it's often referred to as is the fluid mosaic model of membrane structure. It's called fluid because it can change shape, meaning that parts are free to move around, but also because the phospholipids can change position in the same plane. So like so the phospholipids on the top of the bilayer are free to move around as they like. The phospholipids in the bottom of the bilayer are free to move around as they like. So they can move around in the same plane. And then it's referred to as mosaic as the membrane has protein molecules embedded and attached to its surface. And when a cell membrane is viewed through an electron microscope, where we get images or micrographs of cell membranes through electron microscopy, these protein molecules create these regular, kind of almost repeating patterns, or appear to have these regular patterns throughout the membrane, creating this mosaic-like feature to it. So the following diagram just goes through, again, the different components of the cell membrane, coming together to make that fluid mosaic model. So you have the phospholipids that form that bilayer, the cholesterol that is found embedded between those phospholipids and will help regulate that fluidity. And then you can have those integral proteins that are transmembrane and peripheral proteins that can move about or can be associated with integral proteins as well. You have the protein channels that will allow substances to move in or out of the cell. And then you can also have the sugar chains, these long chains of carbohydrates attached to membrane proteins or attached to phospholipids directly. This is generally referred to as the glycocalyx, that overall layer of sugar chains. Individually, a sugar chain attached to a membrane protein would be referred to as a glycoprotein, and a sugar chain attached to a phospholipid would be referred to as a glycolipid. So terminology that will be important to know. And then lastly, you will need to be able to draw a representation of the fluid mosaic model using the following components. And so we will go through in class what this drawing should look like, what shapes you should use to represent each of these different structures. So that is it for this video of 1.4, 1.3.